Thank you to the organizers. My lab works on the Wnt signal transduction pathway, a pathway that's important for the development of all animals. Inhibition of Wnt signaling results in a number of developmental defects in many tissues, where overactivation of Wnt signaling is the initiating event in the onset of colorectal cancer. So the bulk of the work in my lab is in studying known components in the Wnt signaling pathway and in identifying new components and trying to figure out how they function with the idea of understanding both development and disease. But today I'm going to tell you about a different and new story for our lab, and that is in the analysis of Wingless itself as a morphogen. So the idea that morphogens exist, that there are molecules that are synthesized in a subset of cells that are able to be secreted and spread to distant cells where they activate signaling, has been around for more than a century. So the idea here is that at high concentrations of the morphogen, a certain threshold is reached and certain target genes are activated, versus at lower levels, a different subset of target genes are activated. And all of this is important for tissue patterning. But whether morphogens actually exist had been controversial for many decades. And it really wasn't until the mid-90s that there was evidence that proteins spread and could affect tissue patterning, and that came from studies in Drosophila, and Wingless was one of the first to be identified. It was already known at that time that Wingless was critical for patterning of the adult wing. So in the absence of Wingless signaling, you saw severe defects in wing patterning. But linking that to actually morphogen and long-range tissue patterning took the development of a number of tools that came from many labs and ultimately was used by the labs of Gary Struhl and Steve Cohen to test this morphogen hypothesis. One of the critical tools was the establishment of a wingless antibody. So here's this antibody staining a wing imaginal disc, and here is the dorsal ventral boundary of that disc. So you can see high staining of wingless protein in the wingless producing cells and lower levels of protein where Wingless has spread, consistent with the morphogen hypothesis. Second was the identification of target genes, all dependent on Wingless signaling, but some that were high threshold and some lower threshold. So high threshold target genes that were just activated where Wingless was high, near the source, and lower level thresholds that were activated farther away. And then the correlation between spreading and target gene activation high and low level, and the importance of that for tissue patterning. And one of the really critical tools for this testing was the development of a tool in Gary Struhl's lab. So that lab wanted to prevent the spreading of Wingless. And they did that by taking the Wingless protein and membrane tethering it, by inserting a tag from the neurotactin protein, a transmembrane tether. And what they tried then is by overexpressing wild-type Wingless in a clone of cells in the wing disc, they could activate target gene expression, both high-level and low-level, not just in the cells where Wingless was overexpressed, but also far from that source. And in contrast, when they overexpressed the tethered Wingless, they found that, again, high-level and low-level target genes were activated, but only in the immediate neighbors. That evidence suggested that Wingless spreading was indeed critical for long-range signaling and tissue patterning. And it was such a splash at the time that here, after decades, there seemed to be resolution of this controversy that these papers were published in high-profile journals. And this is the last line from the Struhl paper. In our opinion, the present results with Wingless provide a strong argument for accepting the existence of morphogen gradients as fact, and for focusing further analysis on how such gradients arise and how they organize growth and pattern. And this was mid-90s, now we're 20 years later, and that's basically what's been happening. Many groups studying how Wingless spreads, trying to figure that out, and how tissue pattern is organized with that. The group of Jean-Paul Vincent in London, now at the Francis Crick, was one of the groups that was very interested in Wingless spreading. And as a tool to study that, they wanted to block wingless spreading. But now, 20 years later, we have the advent of being able to knock in a mutant into the endogenous locus. So that's what they did. They took this tethered wingless and knocked it into the endogenous wingless locus. So now we had tissues where there was no wild-type wingless, and only tethered wingless was made. And then they asked what happened. This was sort of their baseline for their spreading studies. So again, here's the control, wild-type wingless, high levels at the dorsal ventral boundary by antibody staining, lower levels away. And then the tethered wingless. You can see high accumulation of this protein in the cells, in the source where wingless is made, and very little spreading. So this suggested that the tethering was indeed working, and then they could ask what happened to patterning, fully well knowing the results of this. But they got a really big surprise, which is that the tethered wingless was compatible with normal patterning of the wing. Okay, so all wingless patterning looked nearly normal. The size of the wing was reduced. So they said it was about 12% less than wild type, and the development of the wing was delayed. But the surprise was the patterning looked okay. And then even a bigger surprise was not just the wing looked normal, but the external surface of the fly looked normal. So the legs looked normal, the halteres, the many tissues in which wingless is required looked largely normal. So you can imagine that also made a big splash. That was published in Nature. Now this is 2014, and ends with the line of this abstract. We suggest, therefore, that the spread of wingless is dispensable for patterning. And given this, then now the big Wnt signaling reviews, which are published now in Science and Cell, say wingless only functions at short range. So that's where we are right now in 2018. So it looked like the end of this morphogen model for wingless and tissue patterning, that it was time to let go. That was my reaction to this, after decades of following this, because, it was the fly data that was so instrumental for showing that wingless was a morphogen, and now the fly data was showing the opposite. But the implications are broad for development and for disease states. And so in my lab, we started to think about, it did look like the external surface was normal, but what about those internal organs? They form through different mechanisms, and they have different demands. And we had been studying wingless signaling in the adult gut. It was just a great model for us to test new components and mechanism. So I just want to tell you a little bit about the adult gut and then tell you about our studies. So here's the adult gut as it looks inside the abdomen, foregut, midgut, hindgut. If you take it and stretch it out, you can see foregut, midgut, hindgut, and then different compartments, different subdivisions. So within the midgut, anterior, middle, and posterior midgut. And each of these compartments have different physiological functions, different cell fates, different gene expression patterns, and they're separated by distinct boundaries that prevent mixing of cells from one compartment to another. That was shown by Ellen Spradling's lab. In addition, these boundaries can sometimes have constrictions, sometimes contain valves or sphincters to control the passage of food. And the remarkable thing was when you look at wingless expression in the gut, you can see that it's expressed in the epithelium at all the major compartment boundaries. And that's true not only in the epithelium, but also in the visceral muscle that surrounds that epithelium. Okay, so also we see enrichment of wingless expression at all the compartment boundaries. And then when we look at target gene activation, and this is building on the work of Bruno Lemaitre, and this is one particular target gene called Frizzle3, and here we're staining the gut with Frizzle3, and we can quantitate that expression. And again, we see, like wingless, that the target genes are also expressed at the boundary, peaking at the source where wingless is, and then decreasing as a function of distance. And you can see the target gene expression at all the boundaries, and you can see them going bidirectionally from the source of wingless. So we thought, what a great system to test whether wingless spreading is important. Is it going to be like the wing where it's completely dispensable? The bulk of this talk is going to be focusing on the midgut-hindgut boundary. And that's the boundary between what's the small intestine and the large intestine. And just by simple staining, so here we're just staining with DAPI just to see the nucleus, you can see this dramatic change and boundary at the midgut-hindgut boundary. So here's the midgut. You see these very large nuclei These are from the enterocytes, the absorptive cells in the midgut, okay, for absorbing nutrients. And in contrast, in the hindgut, where water absorption is the key function, small cells, small nuclei. And then you see this transition zone. That's the midgut-hindgut boundary. These structures, the midgut and hindgut, are derived from completely different precursors. So the midgut from endoderm and hindgut from ectoderm. And then because of that, you see different gene expression patterns. So here's just one example, FAST3, low levels in the midgut and very high levels in the hindgut. All of this just helps us in our studies. So the marked differences between midgut, hindgut, and the boundary are seen not only in the epithelium, but also in the muscle. So in the midgut, you see both longitudinal muscle arranged in parallel arrays, circular muscles with a characteristic banding pattern, and in contrast, in the hindgut, you see primarily circular muscles with a different banding pattern, and here's that transition zone. You see thinning of the longitudinal muscles, they reach over the boundary and attach to the hindgut. Here's what wingless expression looks at this boundary. This is a transcriptional reporter GAL4 knocked into the wingless locus, and you see the high expression at the midgut-hindgut boundary, and same with the target genes. Two different ones here, frizzled and naked, high levels at the boundary, and they decrease as a function of distance from the boundary. Okay, now with the setup, we can ask, what happens when you prevent wingless spreading? Here's just looking at the target genes. You see a dramatic reduction in the spatial range. This is frizzled, but it's the same thing with naked. This is not dissimilar from what was seen in the wing disc. There also the spatial range was decreased, but patterning was normal. What about in the midgut-hindgut boundary, so the adult gut? So the most dramatic thing we see, this is about 80 to 90% of the animals, is this loss of the midgut-hindgut boundary. What it looks like is there's a direct joint between the midgut and the hindgut. And we can also see this abnormal constriction at the joint. And this can get quite severe till you see a twisting between those two compartments as if you were wringing out a towel. You can see also defects in the muscle. I'm going to show you this at higher mag. Here's wild type. Here's the tethered wingless. That boundary region is lost. The longitudinal muscles are no longer in parallel arrays. The circular muscles are not bundled properly. And the hindgut muscles are also not patterned properly. I think that might be why the longitudinal muscles are having trouble finding them. This is just to show you another example of what the patterning looks like. Now this is within the compartment. We're standing with myosin and alpha-actinin, and you can see the dramatic disruption, both in the longitudinal fibers where there's now excessive branching and excessive breaks, and in the circular muscles. Just look at the myosin pattern. It's sharp bands here and diffuse and elongated here. I'm going to just turn now to another. So the gut folding is quite similar between animals, and at the midgut-hindgut gut boundary, it's smooth, but just after that, there's a fold that accompanies where a valve is going to be formed. And we can actually see that folding as we take out these animals and fix them. Here's the midgut-hindgut boundary, and then the pylorus, the anterior region of the hindgut, and then the ilium. This is where that fold occurs. With the tethered wingless, we no longer see that. And in fact, this whole region seems to be lost, as if there's a direct joint between midgut and ilium. So we're losing the normal folds, and we're seeing abnormal twists when wingless is not allowed to spread. And here's the epithelium. Okay, normal ordering in the hindgut is lost, the transition zone is lost, and here in the midgut, you can see a markedly increased size of the midgut, markedly increased number of cells. When we look for cell proliferation markers, they're increased, and as is cell death. So severe disruptions in the epithelium too. So this is just a summary of this. We see defects in the epithelium in multiple processes, defects in the muscle, problems with gut folding, and problems in compartment specification when wingless is not allowed to be spread. So then we asked, because there's two effects with this tethered wingless. One is there's decreased signaling or decreased spreading in the cells away from the source, but there's also increased accumulation in the cells that make wingless. And we thought that maybe either one of these, gain of function or loss of function, or both, could be causing these phenotypes of tethering wingless. So we wanted to ask, and we did that either by inhibiting wingless with a large number of ways or overexpressing wingless at the midgut-hindgut boundary. And this is what we see. So this is now overexpressing using a autopromoter, so at the Midgut-Hindgut boundary and into the posterior midgut, an enzyme that is a negative regulator of wingless. So cleaves lipid off of wingless and therefore inactivates this. But we've done this with a number of different tools. And we see similarities to what we see when wingless is tethered. Large posterior midgut, too many cells, loss of this transition zone, and loss of that whole pylorus. The phenotypes here are more dramatic than with the tethered wingless, but are going in the same direction. And even when we're inhibiting wingless in the epithelium, we see defects in the muscle as well. So here, this is the same animals, but you can see the longitudinal muscles are no longer making contacts. They're no longer doing that thinning. The transition zone is lost. The circular muscles are not doing their banding and bundling, and on both sides. So 2 points I just want to leave with this part is that it looks like the tethered wingless is similar to inhibition or reduction in wingless signaling. And the second point is that wingless signaling in the epithelium seems to be critical for patterning in the muscle. And wingless spreading might be important for this process as well. So this is just the last point I want to make. And that we were interested in signaling crosstalk because wingless is not the only signaling pathway that's expressed at this boundary. There's a number of others. When we looked at DPP, so TGF-beta or BMP signaling, that also shows this graded pattern at the Midgut-Hindgut boundary. Here's DPP itself, high levels at the boundary and lower as you get away. The target gene for DPP, thick veins, same. So they're looking like wingless target genes. So we wanted to ask whether DPP was indeed dependent on wingless. So again, here's a wingless target gene, here's a DPP target gene, and what we found is that when we inhibit wingless signaling, we also lose DPP expression, okay? And we did that by making clones of wingless pathway components. So then we asked, wingless is important for DPP itself, what if you prevent the spread of wingless? Is that important also? And this is what we see. So wild-type animals, and this is the DP target gene DAD, versus the tethered animals. And again, you can see this severe reduction in the spatial range of DPP signaling when wingless is not allowed to spread. Okay, so then the last part, and I'm not going to have time to show you this, is that we wanted to ask, is wingless also important at other compartment boundaries, or was this just a unique situation at the Midgut-Hindgut? So we looked at this middle midgut region where there's 2 gradients of wingless that flank the region, and we see qualitatively similar effects, meaning that this is the region in the gut that contains the acid-secreting cells, which are called copper cells. When we lose wingless spreading, those copper cells are largely lost. Their differentiation is disrupted. We see overproliferation of cells just outside this compartment, and we see massive reduction of the middle midgut compartment itself, and severe defects in muscle patterning as well. So it looks like wingless spreading is going to be essential for a number of different compartments in the adult gut. So just to summarize here, here's the range of phenotypes we've seen at both the middle midgut and the Midgut-Hindgut in the epithelium, in muscle, multiple different processes that are important for muscle. And then there are these sort of bigger structures, the sphincters, valves, gut folding, and then signal pathway crosstalk. So, you know, I hope that some of this biology is really new, the phenotypes we're seeing, and I hope can be followed up with as we go forward. So I just want to finish up with the question of, is wingless spreading important or not? The studies in the wing disc were critical, and it looks like yet that the studies in the adult gut are suggesting that wingless spreading is important. And so before we completely let go, you can see that gut and wing are so different, are quite different from each other as far as the wingless gradients. You know, when they're made, how long they persist, how long they are, just how large these structures are. There are large differences. And I'm just curious, as we look more at different contexts and we identify others where wingless spreading might be critical, that might reveal to us core principles of when spreading might be important, whether as opposed to when it's dispensable. Okay, so this work was led by a graduate student in the lab, and this is our gut team. She's helped by Depes, Hasina, and Junghan. I want to thank the many, many groups that have helped us with stocks. This is for our gut project, but so many of you through the years. And also thank to our funding sources. And thank you for your attention just before lunch.